Hello and welcome to this lesson. In this lesson, I'll be explaining about polymerase chain reaction. Polymerase chain reaction, or PCR, is a technique used to make several copies out of a small fragment of DNA or RNA. It's very important to understand the terms from the name of polymerase chain reaction. So, PCR is made up of two words: polymerase, which is the enzyme that makes polymers of any other molecule, which is in this case the DNA, while chain reaction is a type of chemical reaction that progresses in an exponential way. So the first reaction produces 2 molecules, the second reaction produces 4, and the third will make 16 molecules, and so on. So after a few hundred reactions, billions of copies are produced. Before talking about how the process happens, it's very important to understand the things that we need in order to perform PCR. So PCR requires a machine known as thermal cycler, or simply PCR machine. Inside the PCR machine, we have small tubes in which molecules are added to it, and the reaction takes place. So the things we need are tag polymerase, primers, DNA templates, and nucleotides. Tag polymerase is a type of DNA polymerase, and as we know, DNA requires DNA polymerase for replication using existing strands as a template. The DNA polymerase used in PCR is known as tag polymerase after the heat-tolerant bacteria from which was isolated, named Thermus aquaticus. And this bacteria lives in a hot spring, and it's very heat stable, and it is active at 70 degrees. The second important thing that we need to perform PCR are the primers. DNA polymerase cannot initiate the reaction. They need the help of primers. Primers are short sequences of nucleotide, usually 20 nucleotide in length. Primers provide a starting point since DNA polymerase cannot initiate the reaction, but it just propagated. Primers help to choose the exact portion for amplification. In each cycle, we have 2 primers, and primers are designed so that they isolate the targeted region of DNA to be copied. DNA template is a segment of original DNA in which we want to amplify it. And the nucleotides are the building blocks of DNA synthesis. Now, how the process of PCR happens? The process of PCR requires 3 steps: denaturation, annealing, and extension. The first step is known as denaturation. Denaturation occurs when the PCR machine is given a heat up to 96 degrees. So the double-stranded DNA is now separated into 2 single strands because of this heat. Heat breaks hydrogen bonds between the base pairs. The second important step is annealing. Annealing requires cooling down of the PCR machine so that the primers can now bind to its target sequence. The third step is extension. So after annealing, the temperature is then increased to 72 degrees Celsius, in which Taq polymerase is now active. So Taq polymerase can now find the primer and propagate the reaction and replicate the DNA strands. During synthesis or extension, two identical double-stranded DNA molecules are now synthesized. Now the process is repeated for 25 to 30 cycles to amplify billion of copies of target DNA, and this can be happen in 2 to 4 hours. PCR is widely used other than for detecting virus such as coronavirus or HIV virus. It also can be used at a level of crime investigation as a genetic research, like genotyping, cloning to detect any mutation. It can also be used in forensics and to test the paternity, and it can also be used in molecular biology.